Okay, well, look, let's get started on today's EV Club presentation. So, welcome everyone. Great to see you all here today. My name's Andy Hill. I'm from Victoria University here in Melbourne, Australia, where it's evening time. So, it's good to see a great international representation here. And this is a EV Club that's brought to you by a couple of groups to do with ICEV. The first being the EViNS group, which is the extracellular vesicles in the nervous system group, which is a special interest group of the International Society for Extracellular Vesicles. And it's been going for a couple of years now, the special interest group. And you can get further details on the ICEV website. But the idea of the special interest group is to bring together EV researchers working on particular topics. And this group brings together researchers working on anything to do with EVs in the nervous system. As part of the special interest group, we have a number of working groups that are working on some subtopics within that area. And one of those is around EV-based biomarkers of the nervous system. And so that's going to be the topic of the presentation tonight. Also, just to highlight that the EViNS group is having its second conference on EVs in the nervous system, which is being held in Canada from the 29th to the 31st of October this year. You're still able to register for the meeting, so please go to the ICEV website if you're interested in attending that meeting. Also, I'm the editor-in-chief of the Journal of Extracellular Biology, which is where the paper we're going to talk about tonight has been published. The Journal of Extracellular Biology, or JEXBio as we call it, is a sister journal of the Journal of Extracellular Vesicles, and it's also published by ICEV. So today's paper is going to be presented by Claire Bridel, who's from the Translational Biomarker Group in the Faculty of Medicine at the University of Geneva in Switzerland. And the topic is around cerebral spinal fluid derived extracellular vesicles. And the paper describes a proteomic and transcriptomic comparative analysis of enrichment protocols. So it's a great topic for all of those involved in biomarker research and also around standardization and adapting different protocols for various EV research. So welcome, Claire. It's great to see you today. And I'm going to hand over to you now to introduce yourself and go through your presentation. We're going to keep everyone muted until the end when you'll be able to ask some questions. And if you have questions, please feel free to put them in the chat and we'll get to them after the presentation. So Claire, welcome and over to you.
Thank you, Andy. Thank you very much for inviting me to present our work. And hello, everyone. I'm going to start sharing my screen. Let me see if this works. Is that working for all of you?
Perfectly.
Yeah, okay, great. Okay, yeah, so my name is Claire Bridel. I'm a researcher and neurologist at the Faculty of Medicine of the Geneva University. And I'll be presenting this paper on behalf of the whole group. So we are a group of people interested in neurological conditions and ways to diagnose them. And in this sense, we were interested in looking into EVs enriched from CSF to try to see if they could give us some information about pathological mechanisms, or at least molecular changes associated with conditions that are restricted to the central nervous system, with a specific interest in multiple sclerosis. But before diving into that topic and examining the EVs of our patients, we were looking into how we were going to enrich these vesicles. And going through the literature, we found that there were, as you know, no gold standard to enrich these vesicles, and not a lot of information of how the enrichment protocol we would choose would impact the downstream analysis. So mostly proteomic and transcriptomic, but maybe also in the future lipidomic. So this is why we decided to set up this study to compare, like really head to head, as stringently as possible, the different enrichment protocols, and we looked into the literature to find out which were the protocols that were most used, and we found that ultracentrifugation and size exclusion chromatography were the most, at least the most of the papers published used these biochemical means to enrich EVs. We also found a lot of publications using magnetic beads coated with the antibodies, mostly part of kits that you can purchase. But we decided not to include these enrichment protocols because, well, they're kits. You can't really play around with them easily, and also because the beads prevent downstream, well, at least straightforward downstream proteomic analysis. So we set out to compare ultracentrifugation and size exclusion chromatography. So I'm going to show you on the next slide here the experimental setup. So basically working together with a university hospital made it possible for us to have like huge amount of CSF. So basically what we did is that we collected leftover CSF of diagnostic lumbar punctures, centrifuged this CSF immediately, and then froze the supernatant and stored it until D-Day, the day of the experiment. And on that day, we thawed CSF supernatant of multiple patients and pooled them in order to have a homogeneous pool of supernatant. And then we set out to compare 4 different protocols. So we included 2 protocols with size exclusion chromatography, preceded by ultracentrifugation, because we decided to, since we decided to start with really large amounts of CSF, so 7.5 ml CSF per replicate. And so we had, prior to doing the size exclusion chromatography, we had to reduce the volume. So we did an ultrafiltration step before that. And we included 2 different columns. So these are the Eisen columns, the commercially available Eisen columns that are either they're called Sec 35 and Sec 70. Sec 35 is supposed to enrich particles from 35 nanometer diameter to 350 nanometer diameter, whereas the Sec 70 is supposed to enrich particles from 70 nanometer to 1,000 nanometer diameter. So not knowing how this would affect the outcome, we included both. Then we used a standard 2-step ultracentrifugation protocol, and also a combination of ultracentrifugation followed by a size exclusion chromatography. And we also included some controls, so some plain, let's say, supernatant, and also some ultrafiltrated supernatant. And what you're looking at here are the aliquots. So we did 6 replicas per experimental protocol, per EV protocol, EV enrichment protocol. And in green are the samples or the replicas we use for mass spectrometry, in pink for RNA sequencing, in yellow electron microscopy, and blue was Western blotting. So that's the setup. So it was a lot of tubes, almost 300 ml CSF that we really homogenized well. And then we started. And so we really did everything in parallel. And what you're looking at here is the NTA results. So in red, so we ran NTA on all 6 replicas per enrichment protocol. And what you're looking at here in the A panel is the mean particle concentration and particle size. And in the shaded line is the standard deviation of these 6 replicas. And in red, this is the results of the the SEC35 protocol. So you see that that's the one that has the highest particle concentration, almost 1 log more particles than ultracentrifugation in blue, and size exclusion chromatography with the 75 nanometer column. And then down there, you see the combination of ultracentrifugation and size exclusion chromatography. So we figured, okay, if we do both in series, then we'll get like ultra pure, maybe ultra pure vesicles, but maybe, but we were unable to. Well, we lost too many material along the way, and we didn't have enough material to continue with the proteomic and transcriptomic analysis, unfortunately. So we left that protocol quite early on aside and concentrated on comparing the size exclusion chromatography protocols and the ultracentrifugation alone. And what you're looking at here in B is actually the mean particle yield. So you see that there is really the yield of the SEC35 is really well above the others, and this is the size and of the mean size and standard deviation of the particles. And what you see here is that the protocols that include an ultracentrifugation step actually have larger mean particle sizes. We figured maybe because of the deformation of the procedure of the ultracentrifugation, or maybe just some vesicles that fuse. Now we're looking here at some characterizations of these particles. So you see, yeah, so we see that the mean particle size is around 140 nanometers in diameter. So that's pretty similar for the 3 protocols we're going to look at in the next step. So this is the size of a small EVs. The distribution looks a bit like what's been published for plasma. And so here we're looking at the Western blot analysis. So if we concentrate on panel A, you have here the fraction 2 to 10 of our size exclusion. That's the SEC35 protocol. And this is the SEC75 protocol, and this is the ultracentrifugation protocol. So, and if you come back here to panel A, you see that TSG, so a putative EV marker, peaks in fractions 6 and 7, and this is actually also where the particle concentration peaks. So this is where we considered the particles, the EVs, to be, and we pooled these 2 fractions for these 2 protocols and analyzed the pooled fractions in the further experiments. And you see that some markers have a little shifted peak intensity. For example, here CD9 peaks rather in F7 and F8, could mean that either it's a marker of smaller vesicles, or maybe it's just the free protein also that comes out. And you see that albumin, so yeah, sorry, I forget to mention the controls were CSF protein concentration adjusted, and human astrocytes. And you see that albumin is nicely where you would hope it would be, meaning in the last fractions of the size exclusion chromatography, and nicely detected in CSF and not in astrocytes. So it looks pretty much the same, more or less, between these 2 SEC protocols. And here you're looking at the UC pellet, so the ultracentrifugation pellet, where you see that there's an enrichment for putative EV markers and a depletion in the CSF with the same total amount of proteins. And below are some the best images we could get at electron microscopy, which show that we do have bilipid membrane vesicles that are that look like extracellular vesicles. So that was for the characterization. Okay. Now, as I said, for the size exclusion chromatography protocols, we pooled fractions 6 and 7, which were the fractions where we had most, well, really the peak particle count and our putative EV markers. And these results here are results from triplicates. And what you're seeing here are the number of proteins that were detected in all 3 replicas. So we have about 1,400 proteins that were detected in our EV enrichments coming from the SEC35 protocol. And we compared these proteins to those that are detected in ultrafiltrated only CSF supernatant. So as you remember, this was the first step in the enrichment procedure. And you see, interestingly, that there are about 40% proteins that are exclusively identified in the EV enrichments and not in the ultrafiltrated CSF alone. And this is similar to what we see here in the SEC70 enrichment, the output of the SEC70 protocol. We have about 40%, proteins unique to this fraction. The overall number of proteins identified in the ultracentrifugation protocol is higher, so we detect more proteins, but it's all again about 40% that are unique to the EV enrichments. And you see here the overlap between the proteins identified in the EV preparations, and you see that there's quite a nice overlap in the identity of the proteins that are identified in the EV preparation. So just as a reminder, this is really exactly the same input. So there's still some variability. Then we did a gene ontology enrichment analysis of the proteins that are unique to the EV enrichments, so that they were not at all identified in the controls, so ultrafiltrated CSF or CSF for the UC protocol. And interestingly, we found that these proteins were really highly enriched for genes that are implicated in biological processes related to EVs, like the intraciliary transport, cytoskeleton, intracellular transport, phagocytosis, etc. And here I'm just showing you the gene ontology enrichment analysis of the SEC35 output, but quite interestingly, and this is in the paper, the gene ontology enrichment analysis of the 2 other of the SEC70 and the UC looks almost identical. So even though there is a loose overlap in the proteins, that really the biological processes associated with these processes remains really similar. Now we're looking a bit closer at the protein intensity in these mass spectrometry data of exocarta EV markers. So again, here you're looking, it's a bit small, but I hope you can see, we're looking at the triplicates in our proteomic analysis. And for example, here, this is CD9. And you see that the intensity of CD9 is really high in the EV enrichments, whereas it is undetectable or really low, except for one replicate here in the UF in the control. So this indicates that we really have an enrichment by 5 logs of CD9 in our EV fractions or EV enrichments compared to unenriched CSF. And this is nice for CD9. It's also the case for CD61. For some other markers, it's less clear. For example, if you look at HSP90AA1, for example, here the enrichment is less dramatic than for other markers, but overall, we do have a nice enrichment in exocarta EV markers in our EV enrichments compared to the controls. Now we looked at the abundant CSF proteins, that mostly plasmatic proteins that are considered at least contaminants in this enrichment process. And you see, if you look at albumin here, you see that we have a nice depletion in albumin compared to unenriched CSF, and more so in the size exclusion chromatography protocols than in the UC protocols. So this looks nice for albumin. For other, let's say, proteins considered contaminants, it's less clear, for example, HPX here, but overall still kind of a nice, let's say, depletion in abundant CSF proteins compared to EV enrichments. Then we looked at apolipoproteins, and again here some look good, where we have like a depletion in ApoA1 in our EV enrichments. It also looks quite good for ApoE, but for other, for example, clustering, it's not as nice. So this also depends on the protein. And so then we tried to figure out how we could determine which was the enrichment protocol that led to the best, or the highest purity, let's say. So we calculated enrichment scores, which is basically the intensity of a putative EV marker divided by the intensity in the same sample of albumin. So of course this works if we really consider albumin as a contaminant, but it could be that some albumin is bound to EVs. So it has limitations, but we didn't really, we haven't found a better way. Maybe you have an idea as to how to measure the purity of our enrichments. And you see here, this is the mean enrichment score. So for all the markers I've showed you just in the previous slides, and you see that this enrichment score is highest in the SEC35 protocol, and much lower in the SEC70 and UC protocol. So it seems that UC allows to identify a lot of proteins, but a lot of these are probably contaminants in the context. Then we moved to identifying a core proteome. So basically the experiment I'm showing you in this, I've showed you in this manuscript, in this presentation, was repeated in total 4 times with completely independent CSF pools to see how reproducible this was. And we got similar results with all 4 experiments, but in more depth, we wanted to identify the proteins that were present in all 100% or 80% of the replicas. So overall, we have had about, over the 4 experiments, we had about 30 replicas, independent, using different protocols, and we looked at the proteins that were there in 100% of these EV preparations, and not in the controls, so CSF or ultrafiltrated CSF. And you see here that there were 49 proteins that were identified in every single replica. And there was only a small overlap, so only 4 of them were present in the exocarta list. But just to remind you here, we're really looking at the proteins that are only detectable in EV enrichments, and not the ones that are enriched. So of course it's a much more stringent filter. And here you're looking at the 80% core proteome, so the number of proteins that are there in 80% of the replicas, and there we have 87 proteins. And why is this interesting? Well, because maybe this could be this could lead to the identification of markers of CSF EVs, because they are present in a lot of independent experiments. But of course, this remains to be validated. Now we're moving from proteomics to transcriptomics, and we decided, so here you're just looking at one replicate, and we decided to investigate poly-RNA transcripts, because we wanted to see if we could find also some tissue-specific signatures, so transcripts that arise from neurons or central nerve or astrocytes or central nervous system transcripts. And what you're looking at here are the number of transcripts identified in the 3 different protocols. So you see here again, it's a bit of the same trend as with the proteomics. So you have a nice 13,000 proteins transcripts that are common to all 3 protocols. And you see here that there's an additional 10,000 transcripts that are unique to the ultracentrifugation protocol, which could hint at a less specificity of this enrichment protocol again. And quite interestingly, there's only, let's say, about a 50 to 60% overlap between the transcripts identified between 2 enrichment protocols. Again, here we're talking about the exact same input. So there's quite a lot of variability in the individual transcripts that are identified according to the protocol used to enrich EVs. But on the other hand, and that's quite interesting, the gene ontology enrichment analysis, again, here is really very similar between the 3 enrichment protocols. So I'm just showing you the SEC35 results, and that's quite interesting because it shows that even though there's a lot of variability maybe in the individual transcripts, there's still an enrichment for genes that are related to extracellular vesicles, with again the cilium organization at the top. But interestingly, and this is different from the proteomics results, we also have some genes that are tissue specific, so related to regulation of neuron projection or axonogenesis. So this is interesting and different from what we see in the proteomic data. Okay, so conclusions. So what we conclude is that all 3 protocols, so SEC35, SEC70, and UC, enrich particles which have characteristics of small EVs. That the proteome that is unique to the EV preparations and is not identified or not measured in CSF or ultrafiltrated CSF is enriched in genes strongly associated with EV-related biological processes. So it indicates that what we enrich, it's a clue that what we enrich is really related to EVs and therefore contains EVs. And that, on the other hand, the transcriptome is enriched in EV-related biological processes, but also neuronal-related biological processes. And I think it's an interesting difference. And we, as you saw in the very beginning, the EV preparations that resulted from the SEC35 protocol had the highest particle yield. But of course, this is just NTA data. A larger proteome in transcriptome was identified in the preparation resulting from the UC protocol, but the enrichment scores were lower, at least for the proteomic data, which indicates that these enrichments are probably less pure. And what we can conclude is that the enrichment protocol significantly impacts the EV output in terms of individual proteins and transcripts that I identified. You remember that there was about a 50% overlap in the transcripts that were identified in the different protocols. And but however, the Go terms highly associated with the EV preparations are very similar across protocols. So what it could mean, especially in terms of biomarker research, is that individual biomarkers may be not very robust, but on the other hand, like pathway biomarkers may be more robust across experiments. And now for the outlook. So as I said, we had access to a large volume of CSS, which is of course not always the case. So we already did some experiments, and we see that we can go down to at least maybe even lower, but 3 ml CSF input and still get very nice, robust and superimposable results. We would like to have a go at metabolomics of these enrichments. And now we are ready to move forward to look into mechanisms associated with progression in multiple sclerosis when the blood-brain barrier is closed. So looking into really mechanisms that concern the central nervous system, probably using the UF SEC35 protocol, given the data I've just showed you here. And that's it. Thank you. I would like to thank all my collaborators in the group and you for your attention. And I'm happy to take some questions, comments, suggestions.
Okay, well, thank you. Thanks very much, Claire, for a wonderful, very clear presentation on a really carefully conducted study. So we've got time for some questions now. So feel free to put the questions in the chat, or if you'd like to put your hand up, we'll try and get to you in turn as well if you'd like to ask the question yourself. So we've got a question from Bhuvan Bhaskar, who's asking. Sorry, but I did not get why the SEC35 UF protocol have the highest particle yield.
Yeah, that's a good question. And the short answer is I don't know, but I have a hypothesis. So the SEC35 columns enrich for particle, at least that's what they're supposed to do, enrich for particles from 35 nanometer to 350 nanometers in diameter, whereas the SEC70 enriches particles of a larger size. And as you saw, the distribution of the mean diameter of these EVs is around 140. So my hypothesis is that it does a better job. The SEC35 actually does a better job in enriching for small particles than the SEC70 that has a much wider range of enrichment. Do you see what I mean, Bhuvan? I don't know if this is clear.
Yeah, I got it, Captain.
It's a hypothesis, but yeah, I don't have another one to suggest. Yeah.
Yeah, thank you. Thank you for.
Thanks for the question. The next one is from Sinaya Ikezu. Nice presentation. Is there a difference in the particle yield between the first and the second UC steps?
Yeah, well, I don't know. Actually, I didn't test that. Yeah, we didn't test that. We just went for we just tested basically. We just did an NTA after the second. So, but good. Yeah, interesting. Yeah, but yeah, we didn't test.
Yeah, so we do ultracentrifugation. So you ultracentrifuge 7.5 ml, which is a large volume. So I usually do overnight ultracentrifugation for that size of the volume. So I was wondering, if you can complete most of the ultracentrifugation in 2 hours or not. That's my question.
Yeah, no, it's a good question because it would save us some time. And yeah, no, to be tested. Yeah.
Thank you.
Thanks, Sinaya. So Tom Rothstein is asking, what was the UC protocol? Perhaps I missed this.
Oh yeah, no, I didn't mention it. That was 100,000 Gs 2 times with a first certification just to get rid of the debris, but it's 2 times 100,000 Gs. Yeah, I didn't mention.
Thank you.
Yeah.
Lucio Burrell is asking, according to apolipoprotein data, the UF-SEC preparations are enriched in lipoproteins. Is this your conclusion?
Yeah, so this exactly. So I found some data that indicates that apolipoproteins can be bound to EVs in the corona. And this is actually what we think. And it's also what we see in electron microscopy, that we also have some other little vesicles that are not bilipid layered. So yes, it's a hypothesis that actually we do co-enrich apolipoproteins. Yeah, probably. And I think that's probably going to be difficult to prove that, but probably we would, yeah, but that's what we think.
Okay, and the next question is from Anna. I'm not sure if I missed it. Did you find some interactions between proteins as well, i.e. via string analysis? Thanks for the great talk.
No, we didn't do that. Good suggestion. We didn't test that. Yeah. No, we didn't look at that. But
And Flora Chen is asking, nice talk. What kind of normalization was done for the Western blots of the sec fractions?
So we used the entire sec fraction. So what we did is that we air dried every fraction. So we just put everything in, and we measured the concentration of the proteins in another experiment in fractions 6 and 7, and we made sure that our control, be it so it's the ultrafiltrated CSF, had the same total protein amount than fractions 6 and 7 to make sure that the enrichment or depletion in proteins was not due to a different total protein amount. But for the fractions, we put the entire fraction air dried and resuspended in loading buffer, and loaded everything on the gel.
Okay. The next question is from Sindhu Thiagarajan. What was the EV input into the proteomic analysis normalized to? EV numbers or protein concentration?
Was the input in the protein? So here again, we loaded everything, and then you do an adjustment, a normalization for the entire proteome. So the normalization was done at the mass spec step. And again, as for the Western blot, we injected the whole volume. So basically, what came out, so it was about 300 microliters, that came out of the pooled fractions of the second. We injected everything in the mass spectrometer. So, and the normalization takes place afterwards in the mass spec analysis.
Okay. And another question from Bhuvan. What are the major proteins that you found in your proteomics data?
Yeah, we can look at that together. So this we actually published the I think that with the publication you get the protein. So totally unspecific proteins. So we were wondering if we would identify like central nervous system specific proteins, but it's not the case. It's just yeah, non-specific, I would say.
Okay. Lucia Languino, is the proteomic data available? I do not see them in the paper.
Yeah, so that's a very good point. So it's data independent acquisition, and it was really difficult to we didn't manage to upload it on Pride yet. But I'm happy to share it, of course. We're trying to find a way, because you can't, it's less straightforward than with other than DDA. So but I'm very happy to share, and you can just send me an email and we can discuss that.
Yes,
Great.
yeah, thank you. I will. Yeah, I would like to see the results. Thank you.
Yeah, yeah, of course.
And a question from Tom Rothstein. What effect did freeze thaw have, if any? Did you compare fresh with freeze thawed samples?
Yeah, we did that actually. I didn't want to bore you with all that, but we did do. I've got some figures here in the supplementary data. We did do because we tested two things. We tested how freeze-thawing CSF supernatant before enriching affected the output, but we also tested how freeze-thawing enriched EVs affected the output. When I'm talking about the output, I just mean particle concentration and size, because we couldn't do the whole proteomic and transcriptomic analysis, of course, but this would be very interesting. So that's what you're looking at here. So basically this is, no, that's something different. Yeah, it's this one. So if you look at panel A here, this is the distribution of triplicates of fresh. So the number of the particle concentration resulting from an enrichment with the SEC35 protocol done immediately after CSF collection and centrifugation. That's the green one. And the purple lines are triplicates with frozen CSF. And if you look at the concentration that we didn't see that you get after enriching for EVs, it's basically the same. So freezing CSF before enriching for EVs does not affect the particle number. And then we looked at also on the panels on the right is if we let the CSF supernatant in the fridge for 1, 3, 7 days, or if we froze and thawed them in triplicates again, how this affected. You see that it virtually doesn't, none of these differences are significant, so that the concentration does not differ if you do your enrichments immediately after 1, 3, 7 days at 4 degrees, or if you do them after freeze thawing. But of course, this is just, as I said, NTA data, so it's not the whole picture, but it's a first indication that at least it's not, the effect is not drastic. And I think I have, yeah, and this, no. Yeah, that was, yeah, sorry, this was in B, this is, the EVs were all enriched at the same time, but then they were left, sorry, it's the EVs that were left for 1, 3, or 7 days at 4 degrees. So this here was after enrichment. So the fact so keeping your EVs at 7 days in the fridge will affect a bit the concentration, but not significantly. Yeah, sorry for that. Yeah.
Okay, our next question is from ICEV admin. You mentioned minimum volumes as a subject for future study, but what minimum volume would you recommend for proteomics of normal CSF?
Yeah, so in this picture, in this figure here, figure A, you see that, so we looked at 3 different volumes, 10, 7.5, and 5 in triplicates again. And you see that when you go down from 7.5 to 5, there is decrease in the particle yields, but interestingly, as far as the proteomic analysis is concerned, it doesn't affect the number of proteins that I identified. So we went also, it's not here, but we went down to 3 ml, but my impression is that we can go even lower. So I can't answer your question now, but of course, we will be looking at, for example, 1 ml, because I think 1 ml is something that that is accessible to or available in most biobanks, or maybe also in clinical practice. But at least, so when you go down with the input, you reduce the number of particles, but as far as proteomics are concerned, you don't reduce the number of identified proteins. So yeah, so 5, you're still safe, but I think you can go lower.
Okay. It's great to know. Next question is from Marco Falcone. Amazing talk. Maybe I missed this from a previous question, but did you quantify the amount of protein obtained from each fraction?
Yes, we did. I didn't show that. It's true. We did that in separate experiments because we wanted to really we couldn't use, because we're really talking about very, very, very small volumes and an amount of proteins. But we did do that. We did do an experiment that was dedicated to that, where we measured the protein concentration in every single fractions in replicas to see approximately how much protein we had in each fraction. And the highest concentration of proteins is in the last fractions, 8, 9, 10. That's where most of the soluble proteins will end. And then we used this as an approximation for the experiment I've just showed you here for the Western blot analysis, to make sure that we loaded a similar amount of proteins in the controls compared to fraction 6 and 7, to show that there was really an enrichment in EV markers, if you see, and it was not just there was more protein. Is that clear? That's
great. Thank you. We've got a question from Young Hyun Park. Thanks for the nice talk. So SEC35 should enrich smaller vesicles than SEC70, but based on the NTA data, the size distribution of the UF SEC35 and UF SEC70 was similar, although UF SEC35 has a smaller peak at small vesicles, average size about 140 nanometers. Would this mean that most of the EVs in the CSF are around 140 nanometers in diameter?
Yeah, that's my understanding, but it's an interpretation. So my impression is that the SEC35 to 350 nanometer, the SEC35 column does a better job in enriching particles that are in the 150 nanometer range, and that the SEC70, because it is designed to enrich particles with a much broader diameter distribution, basically from 70 to 1,000, it doesn't work as well. But that's just that's just me trying to find an explanation why we have a lower and consistently lower yield of vesicles in the SEC70 compared to the SEC35. So my impression is that the distribution of vesicle sizes is what I'm showing you here in CSF, regardless of the tool you use to enrich for them. But some tools, the SEC35 does a better job in enriching a maximum of these vesicles, probably because it's designed to enrich vesicles that are much closer to the range of the vesicles that you find in CSF. Do you see what I mean? Yeah. But again, it's a hypothesis. I mean, based on what we see. But again, we've repeated this many times, so we're quite confident. We did 4 really big experiments, and we find this consistently. So, yeah.
Okay, that's great. Got another question about the UC protocol. Was the CSF samples simply spun at 100,000 G, or was a lower speed spin used to eliminate cells?
Yeah, there was a lower speed spin before to get rid of the debris. Yeah. Yeah, it was really the standard UC protocol. From the top of my head, I can't remember. Yeah, but there was a first step. So the very first step is the centrifugation. When you get the CSF out of the patient, then get rid of the cells. Then it was frozen, then thawed, and on the day of the experiment, there was again a low-speed centrifugation to get rid of debris, and then you go into the 2 100,000 G ultracentrifugations. Yeah. So yeah.
Okay. Look, we've run out of questions in the chat, so if anyone has any last questions, please start typing out. I've got a couple of questions that I'd love to ask. One is around, you sort of mentioned during the talk about potentially there might be some CSF specific markers. Do you think we might be able to sort of discriminate between CSF EVs and those maybe coming from somewhere else? And would this say something about what kind of cells are releasing EVs into the CSF as well?
Yeah, that's a question I'm very much interested in. Yeah, I was thinking in next experiments to compare, to do an enrichment of CSF and plasma EVs at the same time, and then comparing these proteins that are unique to CSF and maybe unique to plasma enriched EVs, and see if these overlap. So if these are markers of, let's say, EVs compared to the matrix that is around, or if these are really specific to CSF, because of course this would be very interesting because it would allow us to discriminate CSF EVs, potentially EVs derived from central nervous system cells versus the periphery, let's say. So yeah, I would be interested in looking into that. Yeah, absolutely.
And do you have any thoughts around, any differences you might see when you start looking at sort of disease versus, the normal scenario with these protocols, perhaps in terms of yield or,
I don't know,
coverage
because,
of
yeah.
proteome and transcriptome?
Yeah, good question. Well, actually, we don't know what these CSFs, where they come from. They're probably from diseased patients. Well, diseased patients, because, well, by definition, a lumbar puncture was done for clinical reasons. These are not like healthy donors, but it's just a huge, like, it's just, we don't know. Maybe some of them had meningitis, others had probably maybe dementia, others maybe had normal pressure hydrocephalus. So it's a big mix. So I don't know if I have a little bank of CSF of young, healthy individuals, and it would be interesting to compare that. That's also a future plan to see how if because there's some data. You probably know that inflammation, of course, increases the numbers of vesicles that are released. So how pathology affects just EV production and in itself is already interesting. But so I would say we compared 4 different pools of CSF from very different patients, but they were all patients. So I can't compare that for now to the physiological situation. And yeah, that's a good point too. Yeah.
Okay, thank you. And look, my last question, a bit more of a blue sky question, is where is this work leading you in your lab? What are the key burning questions that you're looking to answer at the moment?
Yeah, so we're really interested in a specific neurological condition, multiple sclerosis, and we are interested in, because the mechanisms that underlie later stages of sort of, let's say, the neurodegenerative stages of multiple sclerosis are really unknown, because well, basically you have all we know is based on post-mortem pathology, and the idea now is to use these EVs to try to have them to find out in vivo what's happening in the brain let's say, what are the modifications in the brain of these patients when they degrade, but still in vivo. And so that's the burning question we would like to address. And to that end, we're now collecting CSF from patients who do poorly and others who do age-matched, who do well. And the idea is now to explore, enrich for EVs and look if we find some differences in these 2 extreme phenotypes that could tell us, and then maybe compare that to what we see on post-mortem brain. So yeah, that's the burning question for us. Yeah.
Wonderful. Well, look, we're just got to wrap it up now, but please, everyone, let's join me in thanking Claire for a great presentation and for answering so many questions. And it's great to be able to delve into the sort of technical aspects of studies like this. So thanks so much, Claire, for your presentation today. So thank you
all. And feel free to reach out by email if you have more questions. Yeah.
And thank you to everyone for joining today and for being engaged and asking questions too. And it's great to see you all here today. And please check out the Jexpire website where you'll find this paper, and also if you're interested in the other activities of the EVs in the nervous system group, please check the ISEB website, and we'll see you at the next EV Club. So take care, everyone, and have a great day, evening, morning, wherever you are. Bye for now.